environmental monitoring, and pharmaceutical screening. He has over 85 issued US patents and over 180 peer-reviewed journal publications. He is a fellow member of NAS, IEEE, OSA, RSC, AAAS, and AIMBE. His work has been recognized through the IEEE Sensors Council Technical Achievement Award in 2010, the IEEE Sensors Council Distinguished Lectureship in 2013, and the Engineering in Medicine and Biology Society Technical Achievement Award in 2014. Before we start the session, I would like to say few instructions to be followed by the participants. Participants, kindly post your queries in the chat box. There would be a question and answer session at the end of the webinar. The attendance link will be posted at the end in the chat box. Now, I invite our speaker of the day, Dr. Brian Cunningham, to kindly start the session.
Well, thank you very much for the very warm welcome and your opportunity to meet colleagues in India at St. Joseph's Institute of Technology. And I'd especially like to thank Myra Ganesh, one of the student chapter members who kindly reached out to me and invited me to come and give a seminar to you today. I wish I could come and visit you in person. Here in Champaign, Illinois, just like everyone else in the world, we've been impacted by the coronavirus pandemic, and I've been working at home for almost 2 months now and only seeing people, except for my wife and my daughter, by Zoom. So I'm really pleased to have the chance to meet you, at least by Zoom, and to share with you some of the work that we've been doing in my research group using photonics in the context of diagnostic detection of diseases, and especially focused on cancer and technique that we call liquid biopsy. So with that, let me begin. And first, acknowledge the students and postdoc scholars in my group who are the ones truly working in the lab and doing the things that I'll share with you today. We enjoy financial support from National Science Foundation and the National Institutes of Health. And the work that I'll show you today involves multidisciplinary collaborations with other really wonderful faculty members and friends, Professor Demirci, Professor Smith, Professor Singham-Maneni, Professor Yi Lu, and Manish Kohli are all people working in chemistry, nanoparticle engineering, cancer, clinical care, and microfluidics, that I'm very fortunate to have some really great collaborators to do this multidisciplinary work. And so what I wanted to share with you first is how we approach this interdisciplinary work. And my background as an electrical engineer is in electromagnetics, micro and nano fabrication, sensors, things like that. And to do work in at the leading edge of cancer diagnostics, it's really necessary for engineers like me to find collaborators that do things that complement what I do. And so we've organized ourselves to form something called the Center for Genomic Diagnostics, where I serve as the director of this center. And the idea is developing liquid biopsy techniques to reveal things about human health and disease. And the fundamental direction that we're taking is using information that comes from genome sequencing. And sequencing is something that's becoming more and more common as the price has dropped. Nonetheless, gene sequencing is very labor intensive. And even though it creates a lot of information, interpreting all that information is very difficult. And so our approach, though, is to use genome sequencing to understand things about how individual biology works in terms of cancer genomes, understanding what molecules are produced throughout the body, such as proteins. And genome sequencing is getting so powerful that now you can do genome sequencing on specific tissues or even individual cells. And so gene sequencing generates enormous amounts of information that people with backgrounds in computational genomics can analyze and sort through with the goal of identifying some molecules that we call biomarkers that can become the subject of more frequent, low-cost, non-invasive detection. And so the idea is that by using genome sequencing, we can identify target molecules that can be detected in bodily fluids, even single droplet of blood. And so within a droplet of blood, there's just a wealth of information through the molecules that are in there. Some of them are nucleic acids like microRNA, messenger RNA, circulating tumor DNA. And as I'll describe, some of these molecules can even be carried around the body and protected by something called extracellular vesicles. But we think of liquid biopsy in the context of how it can have clinical impact and guide physicians on how to make decisions about how to treat us with drugs, to diagnose disease, and so on. And so these liquid biopsy tools can be used to understand normal physiology and the effects of aging and nutrition and exercise. There's even biomarkers for stress and anxiety, the effects that we experience in our environment. All will have output that can be measured by molecules in the body through gene expression. But then, of course, biomarkers are also useful for diagnosing disease. And so almost every human disease has biomarkers associated with it, from cancer to cardiac health, inflammation, and of course, pathogens as well. But the information that we gather with all these tests actually feed back to our analysis, and being able to identify biomarkers that have clinically useful information is a big part of our research. And we do this for several purposes. Part can be directed towards medical diagnosis, being able to understand if someone has a disease and what specific type of disease they have. But these are also tools that can be used in pharmaceutical research. For example, understanding the biological effects of a drug, and also tools for just understanding biology at a more fundamental level. So that's what our Center for Genomic Diagnostics is all about. And just to kind of share with you another way, I mean, our team has clinicians, it has engineers like myself, has biologists and biochemists, and basically what we're doing is taking all this information about the genome, the proteome, combining that information with clinical studies, analyzing it all, and then, as I mentioned, identifying some novel biomarkers that can become our targets for detection. So as engineers, we look at those biomarkers and think, well, how can we detect those with better limits of detection? How can we make our detection more specific for one type of molecule and not another? And so this is where the engineers and chemists come in, developing ultra-selective biochemistry methods and ultra-sensitive detection methods that involve microfluidics, involves methods for making our detection able to detect many molecules at the same time, something that we call multiplexing, perhaps detection approaches that can be implemented at the point of care, so perhaps not having to send samples away to a laboratory, and being able to take these technologies and make them simple, easy to use, so they can leave our laboratories and go out and be used by physicians. So in the course of doing all this, we generate a lot of intellectual property. We work with clinicians who can provide us with test samples from their patients, where we can detect molecules in those patient test samples, and then at the end, hopefully develop some clinically validated technologies where we can show how they work in a clinical context, and then perhaps start companies, license our technologies to bigger companies. We're hoping to find ways to have our technologies get used out in the clinical world. And so, as I mentioned, this effort is very multidisciplinary. And so there's many faculty in the Center for Genomic Diagnostics. And I just listed them here. I won't read all their names, but there's a lot of things going on our campus that tie all this together. So we have a cancer center that has a theme called Cancer Measurement Technology and Data Science, that I'm one of the leaders of that. We have an Institute for Genomic Biology where our laboratories are. We have some efforts on our campus to commercialize technology through something called Discovery Partners Institute. We have a very strong College of Engineering, a micro and nanotechnology laboratory where we can fabricate microfluidic devices and many types of sensors. Something called a Healthcare Engineering Systems Center helps link us up with doctors who have interests in health and aging and health disparities. And then we have clinical partnerships with research institutions like a Mayo Clinic in Minnesota, Minneapolis, Minnesota in the USA. So I wanted to share this introduction just to show you that I'm really not doing this alone, that there's a lot of faculty and students and postdocs that are combining their skills to bring you this kind of work. And it's important to be able to work in this multidisciplinary way. So the idea that we're working on is what's called liquid biopsy as a less invasive and painful alternative to the conventional needle biopsy, where instead of extracting tissue and looking at it under a microscope, instead we were taking a few droplets of blood out of a person's finger, then analyzing molecules in that droplet of blood to understand things about the health of the person where the droplet of blood came from. And so the challenge with liquid biopsy is that we have a small volume of test sample. We're not taking a quart of blood out of a person. Instead, in a few droplets, some of the molecules we want to detect are present at very low concentrations. So there may not be very many of these molecules to detect. So it's one of the more challenging problems in biodetection. Liquid biopsy has been in the news quite a lot, at least here in the US. I don't know if you also hear about a failed company called Theranos, had a book and a documentary last year where they raised $100 million of investor funding. At one time, the company's value was $1 billion. And because they had sold the idea of doing liquid biopsy, being able to detect all kinds of proteins and things in a couple droplets of blood. However, since they tried using the conventional technologies to do this and really failed. And there are many lessons to draw from Theranos, but one of them is that new technology approaches are needed to make liquid biopsy a reality. And so there's many different types of molecules in the blood, but one of the reservoirs of molecule information that's very useful are something called exosomes. So exosomes are 30 to 100 nanometer diameter particles that are shed by cells, where the cells expel these exosomes, where the exosome has a lipid bilayer. It has like an outer membrane, just like a cell does, but the exosome takes away some of the contents of the internal part of the cell. So some of the cytoplasm from the cell is carried with the exosome, but the exosome is so small that it can travel throughout the body. And if you take a few droplets of blood out of the circulation, you will find exosomes in it that have proteins, messenger RNA, microRNA that are representative of the source tissue where that cell originated. And so if a cancer cell has mutations in its DNA, those mutations will be found in microRNA molecules that are shed through cells by exosomes. Now, the powerful thing about exosomes is that because they have this lipid shell, it protects the molecule contents inside the exosome from being degraded by enzymes that are found in blood. So if you can capture the exosomes, sort them out, then open them up and analyze their contents, now you have a way for looking for DNA mutations from cells where the exosomes were originally produced. And so what I'd like to share with you today is just one example technology. We're working on several technologies, but I'd like to share with you one story about genomic guided cancer treatment in the context of prostate cancer. There's a technology part of this story, which is how we can use electromagnetics to make small things like nanoparticles look very big, so we can detect each one of them with high signal to noise. And so we have a fancy electromagnetics term for this. We call them photonic plasmonic resonator hybrids. I'll show you what those are. It's actually not a very complicated idea, even though it's a complicated word. But we use this electromagnetics concept to develop a new type of microscope that we call photonic resonator absorption microscopy, or PRAM. And then we can use this new microscope as a diagnostic technology for detecting biomarker molecules. And so we invented a new type of detection assay that we call activate, capture, and digital counting, or AC+DC, that allows us to detect protein or nucleic acid molecules individually and to digitally count them. And so we've used this approach to obtain very low limits of detection with a very simple type of test that can be performed in point-of-care scenarios. Here, we're focusing our attention on detecting molecules called microRNA that are biomarkers for prostate cancer. And so working in collaboration with Dr. Manish Kohli at the Huntsman Cancer Institute in Salt Lake City, Utah, Professor Kohli studies the genomics of prostate cancer. And so the idea is that on the prostate gland, a tumor develops, and those tumor cells carry mutations that allow those cells to proliferate in the form of cancer. And so the cancer cells carry the mutation, but then the cancer cells also produce exosomes that carry microRNA molecules that carry the mutation sequence with them. And so using genomics and genome sequencing, we've identified specific microRNA molecules that have a nucleic acid sequence that corresponds with the presence of a mutation that drives prostate cancer. So it's our goal to detect these specific cancer-related microRNA molecules. MicroRNAs are, as the word micro would imply, they're small molecules. It's a nucleic acid sequence that's around 22 bases long. And then the specific sequence becomes our target for detection because its presence and concentration is related to cancer. So just to show you a little bit about how important knowledge of these microRNA molecules is, this plot shows survival of patients, of prostate cancer patients, as a function of months, where each time this plot goes down by one step represents a person who's passed away from prostate cancer. So what's shown here is a detection of two specific microRNA molecules that have kind of boring names. These microRNAs are called microRNA 375 and microRNA 1290. Each one of those microRNAs has a specific sequence, but when their concentration is high, if you look at this brown curve, when the concentration of those two molecules is high, survival is poor. The patients pass away much more rapidly than if the concentration of those two molecules is low. Those patients experience much longer survival. So just being able to roughly quantify high or low of these two molecules can tell a doctor whether to pursue more aggressive treatment or not for a patient with advanced prostate cancer. We set about collecting blood specimens from prostate cancer patients while they were undergoing chemotherapy. We were interested in whether or not the concentration of these molecules would change as a function of day during the time that a patient is receiving chemotherapy. We are interested in getting feedback about whether the chemotherapy is having an effect on those molecules or not, and whether the tumor might be growing or shrinking without having to have the person undergo imaging every day. And so we had patients put a couple of droplets of blood into this plastic cartridge where the blood takes a spiral path, and then the small objects in that blood droplet, like the exosomes, can travel further along the strip of paper in this cartridge than large objects. And so we found that, oh, did I lose my screen sharing?
Sir, is it okay, sir?
Is it looking okay? It looks
Yes, sir. It's
Yes, sir.
visible.
Yes, sir.
Yes, sir. It's visible.
Okay, good. All right, good. Thank you. So with a couple droplets of blood and put into this cartridge, we can cut out the parts of the cartridge where the exosomes are, and therefore isolate the exosomes from droplet of blood, and then perform genome analysis on that droplet of blood using an approach called real-time polymerase chain reaction analysis. So doing this, we could show that even over the course of 8 days of chemotherapy, the concentration of microRNA 375 could climb by several orders of magnitude. But the patients reacted very differently from each other. Some experienced a large increase, some had more mild increase, in this case only factor of 10. Other patients had their microRNA concentration stay the same or go down. So what we discovered is that these microRNAs change quite a lot during chemotherapy treatment, but that their concentrations can be different by more than 4 orders of magnitude concentration. And so we need to be able to detect over a wide dynamic range. We found that the RT-PCR assay, although it was sensitive, it wasn't sensitive enough. And we also found it was very tedious to do quantitative analysis. And so we sought biomolecule detection method that could meet these requirements. So first, many logs dynamic range, very low limits of detection. In fact, being able to go below 10 to the minus 15 moles per liter, like very low concentration. But at the same time, we wanted this detection method to be simple enough to be performed in a clinical setting, but also to be very specific for the microRNA sequence that we want to detect and not other closely related microRNA sequences. Then also to make this test as simple as possible, we'd like it for it to be just one step. The RT-PCR method involves many steps that have to be performed very precisely for the assay to be successful. We wanted to have a much more simple test. So there's several ways to approach this problem of detecting very low concentration of a molecule. In chemistry, people will use enzymatic amplification, taking one molecule, using enzymes to amplify one molecule into many, and then detecting the many molecules after you've amplified them. A polymerase chain reaction is like this, where you may start with one copy, but then you amplify that one copy into 2, then using enzymes, you can turn that 2 into 4, and so on, so that over the course of maybe an hour, you can have billions of copies, and finally you detect those. Engineers can attack this problem by using confocal microscopes, expensive technology, electron multiplied CCD cameras that have to be cooled to 77 degrees Kelvin. So definitely possible for engineers to drive detection limits down using expensive technology. But what we were seeking to do was to use, to increase the amount of signal that we get for each molecule using nanoparticle tags. And what I'll focus on today is how we can use gold nanoparticles to amplify our detection and enable us to get down to seeing and counting individual molecules. So, our goal here, microRNA detection that can be performed frequently, even on a daily basis, to monitor cancer patients' treatment during therapy and see whether that chemotherapy is having the desired effect on molecules. Okay, so if I were to summarize what it is we're trying to do, we're trying to take the energy from big things like LEDs and lasers that have a lot of intensity, but to concentrate the energy into very, very small volume, such as into a gold nanoparticle who might have a diameter of around only 100 nanometers. So a gold nanoparticle, not much larger than a protein, for example, we're trying to just pack a lot of electromagnetic energy into a very small volume. The tool that we use to do that is something called a photonic crystal surface. The basic job of the photonic crystal in our work is that it's a nanostructured surface that can capture light from an external light source, such as an LED or a laser, and to take the energy from that illumination source and to concentrate its energy in a very small volume on a surface. So that surface can carry really big electromagnetic intensity, but then we want to further concentrate the energy into nanoparticles. And so what's shown here are some gold nanostars that when they come into contact with the photonic crystal that's itself carrying a lot of electromagnetic energy, we want to channel that energy into the gold nanoparticles. So that overall, that's how we make a bridge between the macro world and the nano world, using this photonic crystal as an intermediary. So we can make these photonic crystals on silicon wafers or glass wafers and chop them down into small die. This whole surface here is a photonic crystal, where if we were to zoom in, the photonic crystal has a periodic arrangement of lines and spaces that's smaller than the wavelength of light. In this case, has a period of around 450 nanometers. And here you can see some metal nanoparticles just sprinkled on the photonic crystal. You kind of get the idea that these are around the same size as molecules, maybe a little bigger, but then we can put them on the photonic crystal surface. So just to give you some size perspective, a virus particle around 100 nanometers diameter, one antibody around 10 nanometers size, one DNA molecule around 1 nanometer in terms of its spatial dimension. Now, the property that these photonic crystals have, what we designed them for, is to be an optical resonator. So when we design the photonic crystal, notice that we have a low refractive index material, such as silicon oxide, which is glass. It has a periodic surface structure that's etched into it. Then on top of that, we put a higher refractive index material like titanium oxide. We choose the period, the refractive indices, the dimensions of this grating in such a way that this structure will form a electromagnetic standing wave with one specific wavelength of light that we call the resonance wavelength. So at the resonance wavelength, this photonic crystal will behave as a nearly perfect mirror. So all the light at that one wavelength will be reflected, but for all non-resonance wavelengths, the light can pass right through as if the photonic crystal wasn't there at all. So something special is happening at this resonance wavelength. And so what occurs is that the light enters the photonic crystal, but then gets directed to try to travel sideways. But as the light tries to travel laterally, it experiences this periodic surface structure that back reflects it onto itself. And so within a few periods of the photonic crystal, this structure forms an electromagnetic standing wave that has much higher electromagnetic intensity than an ordinary non-structured surface would have. So we call this a photonic crystal guided resonance, or a PCGR, where at that resonance wavelength, we have hundreds times greater electric field magnitude and optical power than you would otherwise have. Now, we know what wavelength that this photonic crystal resonates. We designed them to be in the red part of the spectrum, around 630 nanometers, as shown here. Now, we can also choose a gold nanoparticle that has a surface plasmon resonance associated with it, where the electrons in the gold nanoparticle will oscillate at a specific frequency. So here's our entire trick, and it's a very easy trick, actually. We simply choose the gold nanoparticle to have a plasmon resonance at the same wavelength that the photonic crystal has this photonic crystal guided resonance. So when we choose those to be the same wavelength, now when we put the nanoparticle on the photonic crystal, it can just suck up energy from that photonic crystal standing wave and then channel it into the nanoparticle. It's almost like channeling energy into a nano antenna. So that's what we call a plasmonic-photonic hybrid. We have a dielectric resonator, the photonic crystal, that couples energy into a plasmonic resonator, in this case, a gold nanoparticle. So we've done a lot of work to understand how these two nano objects interact with each other using electromagnetics computer simulations. And we can show, for example, on a gold nanorod on a photonic crystal, just how large electric field magnitude you can obtain in and around the surface of that gold nanoparticle. Here, the E field squared, actually representing optical power, is 10 to the fourth higher than it would ordinarily be. You can imagine if you have electrons oscillating inside this gold, you can heat it up. You can actually measure higher temperature in a gold nanoparticle that's being excited in this way. But one thing I wanted to really focus your attention on is something that we observed, which is that ordinarily the photonic crystal would be a very efficient reflector at the resonance wavelength. But as we went about absorbing nanoparticles on the photonic crystal, we observed that the photonic crystal went from being highly reflecting to being less highly reflecting. And so we could measure this just by looking at the photonic crystal's reflection with the spectrometer, and then you'll see this effect. So basically, our hypothesis was that when we put nanoparticles on the photonic crystal and they resonate with it, they're stealing energy from the photonic crystal resonance and thereby reducing the reflection intensity. And so again, we did careful measurements. We recently published this in Nano Letters, where we measured the absorption efficiency of gold nanoparticles on a photonic crystal. And what we discovered is that the absorption efficiency of gold nanoparticles actually becomes many times greater when we have this plasmonic-photonic resonator coupling effect. And so just by looking at the absorption as a function of incident angle for a fixed wavelength, we were able to show this enhanced absorption effect. And the basic idea here is that these gold nanoparticles are small. They're only 100 nanometers or so in diameter. But by using this coupling effect, we could make the nanoparticles absorb as though they were particles that were much bigger. So it's really like taking something that's small, making it look big by having this enhanced absorption effect. So we thought about how we could use this as a detection method. So imagine we take our photonic crystal. It has this high electric field intensity region on top of it that we call the evanescent field. That's the electromagnetic standing wave. And then when the photonic crystal doesn't have anything on it, it has this narrow reflection peak, high reflection peak intensity at the resonance wavelength. In this case, it's 625 nanometers wavelength. But now let's imagine we take a gold nanoparticle, just one, and we put it on there. What happens? So just at the place where this gold nanoparticle has landed, it's subtracting energy away. It's absorbing energy from the photonic crystal. And just at that one place, the reflection efficiency will go from high to low because of the absorption that's induced by that nanoparticle. We call this a change in the peak intensity value, and something we can actually measure. We can look at the photonic crystal point by point and see how each gold nanoparticle can reduce the reflected intensity at the place where it's located. So we used this idea to develop a new type of microscope. This is what I called the Photonic Resonator Absorption Microscope, or the PRAM. It's essentially an ordinary bright field microscope where for illumination, we use an LED, so a low power LED whose emission wavelength is around the same as the photonic crystal resonance wavelength. So with the LED, we illuminate the photonic crystal from below, and we look at the reflection coming back through a microscope objective, and we put that reflection into a spectrometer that allows us to measure the reflection spectrum like I showed you. Here's what it looks like in the laboratory. So for the illumination from the LED, we actually have the illumination in the form of a line that extends across the width of the photonic crystal. And so in each point along the line has a spectrum associated with it. And in one snapshot into our spectrometer, we can actually measure 512 spectra, each spectra from a different point along this line. So our microscopy is actually a line scanning method where we move the photonic crystal under the line in small increments to generate thousands of spectra, but each of those spectra represents a distinct place on the photonic crystal surface. So we can analyze all those spectra, determine the peak intensity value, and make a map that it shows us here, every dark spot represents a place where there's a gold nanoparticle attached, and we measure a small reduction in the photonic crystal reflection intensity. So using that as a microscopy approach, working with chemistry professor Yi Lu and postdoc biochemist Taylor Kennedy, we developed this detection assay that we call the Activate, Capture, and Digital Counting, specifically designed for detecting microRNA for cancer diagnostics. So we start with our photonic crystal that we prepare with nucleic acid sequence represented by the sequence of green dots here. Each of these little dots represents one nucleic acid base. I'll describe how we choose those bases in just a minute, but kind of keep in mind that they have a green color here in my diagram. Now we take our gold nanoparticle and we prepare them with a specially engineered nucleic acid that's called a toehold probe. This toehold probe is a molecule that's designed to specifically recognize and capture the target microRNA sequence that we want to detect. So for example, that microRNA 375 that I told you about. There's some interesting chemistry going on here in which when the target and the toehold probe come in contact with each other, the microRNA will actually displace away this part of the molecule called the protector. So what is the protector protecting? Here's a green nucleic acid sequence that's a complementary match to the nucleic acid sequence that's on our photonic crystal. So when the protector is protecting, this gold nanoparticle is not capable of making an attachment to our biosensor. But instead, when the target microRNA molecule is present, this chemical interaction called strand displacement occurs. The protector will go away, and now this nucleic acid sequence in green will newly be exposed. So the presence of one target microRNA molecule will unprotect one toehold probe, therefore making this gold nanoparticle able to be captured on the photonic crystal surface through this interaction of bases, the complement on the photonic crystal to the green sequence that's in that toehold probe. So in this way, we can first activate the nanoparticle, then capture it on the photonic crystal, then using our PRAM microscope, digitally count them. So in essence, what we've created is an approach in which one microRNA molecule results in the capture of one gold nanoparticle. Okay, I put this slide here just to say a little bit more about the biochemistry. It's very interesting. And so these, using like thermodynamic principles, you design these toehold probes so that this interaction occurs spontaneously at room temperature. So no heating, no thermal cycles, nothing required. So notice that all we do is introduce our test sample with the toehold probe gold nanoparticle like a reagent, and then just wait as we capture the activated nanoparticles on the photonic crystal. So this photonic plasmonic hybrid is actually very powerful at giving us high signal to noise. That the presence of one gold nanoparticle results in the reflection going from almost 100% down to around 20%. And so we can see each of these gold nanoparticles very clearly with our microscope, and we can take images with our microscope even at 10-second intervals if we want to. So each of these little spikes actually represents one detected gold nanoparticle. And so using some image processing approaches, we can go and count those and then determine how many molecules we detected. So I wanted to share with you just some of the results from this approach for detecting microRNA 375. This is our prostate cancer biomarker molecule. What's shown here are all PRAM images for different concentrations and at different times during the detection. So if I draw your attention here to 10 femtomolar, after 1 minute, we haven't detected any or very many molecules yet, but you see that we accumulate them after your 30 minutes, 60 minutes, 90 minutes, and so on. More time results in capturing of more gold nanoparticles. Also, if I focus your attention on this row at 30 minutes, going from low concentration to high concentration, you can see as we go from lower to higher, we're detecting more particles. So this is what the images actually look like. Each of these little spots represents one particle. Then we can count them and make quantitative graphs like this that show the particle count as a function of concentration for this molecule. Here is the graph for the 2-hour time point. So we can take that data and plot it as a function of time and concentration, so shown here on the bar graphs. I wanted to draw your attention to a couple of things. One is this gray bar on the left represents the case in which we don't have any microRNA target present. So all the gold nanoparticles are present, and a few land on the photonic crystal and are counted just because they land there by gravity, but not very many. Also, we can go in and change the microRNA that we detect by one base. But then if we use one base and use our biggest concentration, in this case 1 picomolar, still we're not detecting it. So this just shows that the biochemistry is very selective for detecting our target molecule and not any other ones. So we've shown this even no matter where you place the mismatched nucleic acid, the selectivity is still just as good. So I noticed that I want to make sure we have time for questions. So I want to share with you just one more thing quickly, and then I'll stop and be happy to answer questions. We could use this activate, capture, and digital counting approach not only for microRNA, we've also used it for detecting proteins in blood as well. And so in this case, we can prepare our gold nanoparticles with antibodies that specifically recognize a target molecule. In this case, we put our photonic crystal biosensor into a microfluidic cartridge in which we put in 1 droplet of serum, goes into the inlet, and then the serum gets drawn past the photonic crystal by capillary action. And we have an absorbing paper pad at the exit that draws the sample through, where a 40 microliter droplet of liquid goes through in around 30 minutes. So we published this just recently in the journal Lab on a Chip, where our photonic crystal has an active region with a second antibody to bind the activated gold nanoparticles. And then it has a reference area that has a blocking reagent on it, and hopefully no nanoparticles get captured there. But then we use the same PRAM microscope detection approach to count them. Here just shows how we integrated the photonic crystal into this plastic-based microfluidic cartridge that has the absorbing pad in it. So we line these up and assemble them in our laboratory. The inlet is here, photonic crystal is there, and there's the absorbing pad. And we use this in the context of a HIV viral load monitoring assay for detecting a protein called P24. So we prepared our gold nanoparticles with antibodies to P24, and then we prepared the photonic crystal with a secondary antibody to P24. So those two antibodies bind to different places on the P24 molecule. But this just shows the detection of P24 through the gold nanoparticles as a function of P24 concentration on the active part of the sensor and on the reference part. And then we just mathematically subtract the reference sensor from the active sensor to generate a plot like this. And then we've done the same thing for just detecting this molecule spiked into 100% human serum, and then doing this test in only 30 minutes. So the good thing about this approach is that you just add the test sample to a vial that has the nanoparticles in it, and then let them mix for 5 minutes, then dispense them into the microfluidic cartridge, gets drawn through, and then 30 minutes later, read out the sensor, and then the test is done. So just a single step on the part of the user. So I put some other things here into my presentation in case I had extra time, but I think I spoke too long. So I just wanted to mention that we're using other types of nanoparticles, such as quantum dots, as a alternative to gold nanoparticles. And we can use the photonic crystal to also enhance the excitation of quantum dots. So when the quantum dot absorbs light from the photonic crystal, it'll give off more photons than it otherwise would. And then we can do the same type of assay for detecting a target microRNA. But then using the light emission from the quantum dots, here's is shown one individual quantum dot on the photonic crystal. We can detect individual ones, even see them blinking on and off, like the kind of looks like looking at stars. And then we can also perform microRNA detection using quantum dot tags with very similar sensitivity to the gold nanoparticle tags that I showed you. So here, each little bright spot represents one captured quantum dot going down to 0.1 femtomolar, and then counting quantum dots, we can make a dose response curve for detecting microRNA. So with that, hopefully I convinced you that team science approach, multidisciplinary collaboration is very important for bringing together people to tackle really challenging problems like this, where hopefully you could see how a combination of novel electromagnetics, some novel nucleic acid chemistry, like those toehold probes, combined with someone like Manish Kohli, who could tell us about how we can detect certain molecules, have an impact on the way he treats cancer patients, bringing all those people together, very important. Also, yeah, I should also mention some novel nanoparticle engineering going on here also with designing these nano stars, quantum dots to have the properties that we want for getting high signal to noise detection. So with that, really appreciate your attention today, and I'm very happy to answer any questions.
Thank you, sir, for an invigorating session. It was extremely informative. So shall we move into the question and answer session? So shall we move into the question and answer session? Hello, sir. Can you hear me?
Yes, please. Yes, I'm ready.
Yes, sir. The first question is it possible to measure glucose level in body using saliva so that it would be an non-invasive method? And does any instrument exist for that?
Well, glucose detection is like a very interesting problem, of course, because of the prevalence of diabetes. I'm not aware of a saliva glucose sensor, but at least not a commercially available one, but I'm certain that someone has studied that, because of course, pricking the finger is painful and a little bit invasive. And usually detecting from saliva is challenging because of other things that are in saliva and variability from person to person and time of day. And based on like diet and what people have eaten recently. So I wouldn't be surprised if you couldn't find research papers where someone has studied that. But still, at this point, detecting glucose in blood is much more reliable and consistent from person to person. If that information is going to be used quantitatively to guide the treatment of a diabetic person. For example, telling them when to take insulin.
Yes, sir. The second question is, have you tested saliva sample for identification mRNA sequence for different diseases?
So once again, it is possible to find exosomes in saliva. So saliva is usually not a specimen that's gathered in cancer treatment. And so we have samples that all come from serum that come from a few droplets of blood. I think that, again, the problem with saliva is that the enzymes and so on that are in saliva tend to break down molecules and cell membranes. And so I think that saliva may not be like a medium that would, like you would find your exosomes in that haven't been, damaged in some way.
Yes, sir. Okay, the third question is, how do you design nano antennas and what are the materials used to fabricate?
So in our case, we used nanoantennas that are made out of gold, but you can also make plasmonic nanoantennas out of silver. I'm not a nanoparticle specialist, but I know a little bit. I mean, the nanoantennas are like formulated and made like by chemistry methods, but it's possible to have like spherical nanoparticles, rod-shaped ones, star-shaped ones, and so on. So we chose to use gold ones here because gold is not chemically reactive, where like silver would have some of the same plasmonic properties, but silver tends to oxidize in aqueous media. We're also investigating, I didn't talk about it in my presentation, about using magnetic nanoparticles so that we can use externally applied magnetic fields to draw the nanoparticles to the biosensor surface. So in that case, my collaborator, Srikanth Singamaneni, he formulates and chemically synthesizes magnetic plasmonic nanoparticles that incorporate nickel and iron. So they're made by chemical reactions where, he has like reagents that he mixes and heats to precise temperatures and times to grow them to a specific chemical composition and a specific size. And through the chemical reactions and by timing them correctly, he can control those really precisely. I hope that answers your question.
Yes, sir. Thank you. The next question is, what are the side effects of biopsy?
So conventional biopsy involves you're taking a needle that has a hollow core that is like wide. so the hollow core, I think maybe even like millimeter in diameter. So basically the needle is pushed into the tissue, then the needle is extracted, bringing a cylindrical core of material with it. And so you could imagine that needle, because of its size, would damage the tissue. There can be bleeding. There's possibility for infection. And because of just the nature of some tumors, whether they're in the breast or the prostate or the brain, they may be places where you wouldn't want to put a large needle. It's risky. And so may not even want to repeat that more than once. And so that's part of the power of the liquid biopsy method, is that it's not invasive the same way or as risky the same way as a needle biopsy is.
Oh, yes, sir. It was a perfect answer. The last question is, what is the material used in bypass surgery and development of genes?
Oh, in bypass surgery? I'm not sure. Yes. I understood.
Bypass surgery, sir.
I don't know very much about bypass surgery. I'm sorry.
Oh, okay, sir. Okay, sir. Thank you, sir, for answering the questions promptly.
And any other ones?
No, sir. That's the end of the question answer session.
Okay.
Now it's the time to say thanks to all on behalf of the Department of ECE, St. Joseph's Institute of Technology, St. Joseph's Group of Institutions. Information is the lifeblood of medicine, and information technology is destined to be the circulatory system for that information. First and foremost, I thank our guest speaker, Dr. Brian T. Cunningham, Donald Bigger Willett Professor in Engineering, University of Illinois, Urbana-Champaign, USA, for his inspiring speech on finding needles in a haystack. Thank you, sir, for finding time and giving information on very new technologies like liquid biopsy, genomics guided cancer treatment, photonic resonator absorption microscope, photoplasmic resonator, and microRNAs despite your busy schedule. My heartfelt thanks to our beloved chairman, sir, Dr. B. Babu Manoharan, for his consistent upkeep in conduction of this webinar. My sincere thanks to our Managing Director, Ma'am, Mrs. B. Jessie Priya, and our Director, Sir, Mr. B. Sasi Shegar, for their guidance in conduction of this webinar. I would like to express my gratitude to our Principal, Sir, Dr. P. Ravichandran, for his support to conduct this webinar. I also thank our Head of the Department and IEEE Chapter Advisor, Dr. C. Jnana Kausalya Ma'am, and all the other faculties for organizing this event successfully. Finally, I thank all the students, faculties, research scholars from all over India and other countries for their active participation in this webinar. Thank you all. Our next webinar is by an outstanding speaker, Dr. Minglin, who will be giving an interesting talk on From Learning-Based Traffic Reconstruction to Autonomous Driving, tomorrow at 11 a.m. Interested participants, kindly register in the same link and attend the session. Thank you all. Thank you, sir.
Thank you very much. It's very nice to meet you all, and I wish you